Thanks a lot, Ilya, for this very kind introduction, and welcome everyone to my short presentation about generative methods with focus on drug-like molecules with graph-to-sequence neural networks. So this project was part of my PhD, which we conducted in collaboration with Roche, and we have the preprint out since 6 months now. So it's one completed project within industry and an academic fashion. So I'm going to talk about generative methods, specifically language models, and these. So chemical language models. These methods, they work on smile strings. So smile strings are a string representation of chemical structures. There are other string-based representations such as selfies, but smiles, such as the one shown here, are the most prevalent in the field still. And the methods which are used to learn on such representations, I think most are familiar with these methods, are either recurrent neural network, where one learns sequentially on the string-based representation, or graph neural networks, where one learns with positional encoding on a graph representation of the smile string. And so these chemical language models, they are used to generate novel molecules with desired properties, and this is what this project was about. So chemical language models, they are often used in a two-step fashion, where a pre-training is conducted in a first setup on a larger data set of let's say, drug-like molecules, for example, and then a fine-tuning is applied in a second step. For example, if one wants kinase-specific molecules, then one uses a fine-tuning set for using kinase modulators. So there's always a two-step process while they're being applied. And during the pre-training, if we imagine the drug-like chemical space being those blue dots, each of the blue dots represents one molecule, then during pre-training, the model essentially learns to construct these drug-like molecules and therefore learns the structural distribution of these molecules. But it never learns, for example, if two molecules look structurally different, but share, let's say, a similar target or the same target, the model never learns that these two molecules modulate a similar target. And this was the problem that we wanted to address. So there's this fine-tuning approach where one uses a data set to fine-tune the model, but often in industry, if we have one starting point, let's say one ligand or a patent with one scaffold and multiple ligands around this scaffold or one structure, there's not enough data to fine-tune. So what we aimed for was a zero-shot chemical language model, so a model that can be applied in a zero-shot fashion, having one ligand or one protein structure. We wanted to have a model which can be applied for both, so structure-based design as well as ligand-based design, and we wanted the model to incorporate physicochemical properties, let's say number of rings, number of hydrogen acceptors, hydrogen bond donors, because then often depends on which cell type we want to achieve or which properties the initial library should have. We would like to constrain this initially in the input and not afterwards filter the generated library. And to achieve this, we introduced the concept of the drug target intactome, here called like this, where the molecules, if we imagine the chemical space being represented by this graph, the blue dots represent molecules and the orange dots here represent protein structures, and we wanted to connect, let's say, if these two dots share a structure, so share a protein structure, but look structurally different, we wanted the model to explicitly learn this target similarity. And if we look at the data set, so if we build this graph from Campbell, one of the most prevalent data sets for drug-like applications, then we have around 500,000 affinities and 3,000 targets from a ligand-based fashion. And if we only take crystal structures, so proteins which have a elucidated structure, we have 260,000 affinities and 700 structures. So this was the training data set. And the goal was then to train a model which takes one molecule as an input and generates molecules which share the similar bioaffinity with the input molecule, or use a protein binding site as an input and generate the molecules which bind to this binding site. And for that purpose, we designed a graph-to-sequence model So there are multiple approaches how one can tackle this with sequence to sequence or graph to graph. But since we, so two aspects to this, since we wanted to encode the 3D structure of the protein, a graph was more straightforward to implement. And since we wanted to generate simple drug-like molecules, a chemical language model seems to be most suited for the decoder part. So the graph is encoded using 2D and 3D message passing, 3D for the protein structure, 2D for the ligand. Some attention-based pooling is used to pool the node features to a molecular features. Molecular properties are optionally used as a second input, so we looked into both aspects. And then a molecule is generated using an LSTM. So we built such a network based on Campbell, trained it, and looked, let's say, validated two aspects of it. On one hand, how well do the properties reflect, which we define in the input, how well do the molecules that are generated reflect the properties that we define in the input. And if we look at this, so there are 6 properties that we defined during training. So molecular weight, rotatable bonds, hydrogen bond acceptors, hydrogen bond donors, polar surface area, and log P. And on the x-axis, you have the desired property in the input, and on the y-axis, you have the generated molecules, and this is for a few thousand sampled structures. And what we saw is for all of those 6 properties, we have correlation coefficients above 0.95, meaning the properties which we want in the input are with high accuracy reflected in the molecules that are generated. So this was one part that we wanted to validate after training these models. And the second part, the second validation, and the second part that we want to validate, it's first focus on the left side. So we wanted that the model generates molecules that share bioactivity with the input molecule. Let's say if I use a ligand as an input without any structural constraints, I would like that the molecules which are generated have a similar bioactivity than the input molecules. Or it can also be an input protein structure. And if we look here on the left side, so if we use p-par gamma as an input, so it's left out of training. It's in the test set. So we used in total 20 proteins in the test set. So p-par gamma has not been seen by the model, and we use it as an input. And we calculate the fraction of molecules during training of molecules that are generated and predicted to be active on that target. We see that, okay, we use p-par gamma input. We see the model generates a certain fraction for 3 p-par gamma proteins, which are predicted to be active. On the other hand, if we predict the activity on the same input with p-par gamma on Janus kinase, we see, okay, the model does not generate any active molecules, any predicted active molecules on the kinase, but it generates molecules which share some structural features which are known for p-par gamma. On the other side, if we use the Janus kinase as an input, the model doesn't generate any molecules that are predicted to be active on p-par, but generates a large fraction of molecules that exhibit structural features which are known to be active on the Janus kinase. And we benchmark this in comparison to a fine-tuned RNN with fine-tuning the RNN on 5 small molecules for 20 targets. And if we look at this plot, so we compared then the fraction of molecules that are generated by the different models to be active for these 20 targets. And if we are above here on the y-axis, if we are above 0, means that the model that we introduce outperformed the RNN, whereas where we are below 0 means that the fine-tuned RNN outperformed the zero-shot model. And what we saw is that so both generate molecules approximately in the same ballpark, but for 16 or let's say about this statistical significance, for 15 out of the 20, the zero-shot model outperforms the fine-tuned RNN for those targets. So we could show that we have a zero-shot application which incorporates properties with high accuracy, and we can also reflect bioaffinity of the generated molecules. So having this model in hand, we wanted then to prospectively apply this. So we used the same training procedure by leaving out these 20 targets shown before during training, and we applied it to one of the targets, namely p-par gamma. So we used the p-par gamma co-crystal structure as an input, so without any ligand, just the binding pocket, generated a library of 300,000 predicted for these 300,000 molecules the bioactivity using known structures, and defined a novelty score based on scaffold and structural features, since we didn't want to re-identify known PPAR modulators. So we want to be in the top right, as novel as possible, but still exhibiting known features to a certain extent that we can, with a certain accuracy, say that they are active. So we want to be in this space on the top right. And therefore, two ranking procedures were applied, one for PPAR gamma and PPAR delta, so with the goal to identify a dual modulator, and one only based on PPAR gamma, so only based on the activity on a single target. And what we saw is that, so these are the molecules which were then synthesized, named compound 1 with the goal of being active on PPAR gamma and PPAR delta, and we could show then after synthesis, the molecules were tested in a binding affinity assay, that compound 1 was active on both of those targets, whereas compound 2, which was the de novo design, and compound 3, a regio isomer of it, since it was during the synthesis, this was a free to isolate. It has been isolated to and tested, and for those two molecules, as predicted by the model, we could observe a single activity on PPAR gamma and no activity on PPAR delta. So the initial hypothesis that we can de novo design for dual modulation and single modulation of this target was could be confirmed. And let's say for those two, so n equals 2 molecules, of course one would like to do 100 molecules if possible, but synthesis was also in this project the most limiting aspect, so we were only able to do 2, or in this case 3 molecules. And afterwards we showed on 3 different nuclear hormone receptor targets, so the family where PPAR belongs to, that these molecules are all inactive on other nuclear hormone receptor targets such as LXR, RAR, and RXR. And we could also show that they are non-toxic, so up to a concentration of 50 micromolar, we observed no cell death on HEC 293 cells. So we could show that they are selective, exhibit their desired activity, and are also non-toxic. And as a final experiment, we co-crystallized one of the designed ligands with PPAR gamma and could show that the expected binding mode with the 4 key hydrogen bond interactions in the pocket is observed by the binding pose during co-crystallization. With this, I sum up. So this is currently still a preprint. I hope not that long anymore. It's available. Code is also available. And what I introduced was a drug target interactome concept for structure and ligand-based design in a zero-shot fashion. We compared this with RNNs and showed similar performance for 20 targets compared to fine-tuning. And we introduced or demonstrated a prospective application on PPAR gamma and PPAR delta. With this I come to the most important slide. So this was a highly collaborative effort, since experimental, so biology and chemistry was conducted in combination with computational chemistry. So there was a big team. Well, most likely I would thank the collaborators at Giesbert Schneider's group at ETH Zurich, and also our collaborators at Roche. With this, thanks a lot for your attention.